Okay, here we go with the genetic code. This is how we figure out what DNA is trying to say to us. What are you saying? Okay, so how are the instructions for assembling amino acids into proteins encoded into the DNA? Again, what are you saying? So 20 amino acids, but only 4 nucleotides in DNA. So they can't correspond 1 for 1. They can't even correspond 2 for 1. So how many nucleotides is it that correspond to an amino acid? And the answer is 3. Okay. So the flow of information is in a triplet code, a series of non-overlapping, that's important, 3 nucleotide words. Okay. ATG means something. Okay, it's 3 nucleotides. And as I said in the previous screencast, we call these every 3 nucleotides a codon. I think we're going to get to that in a minute anyway. Okay, so these words in DNA are transcribed into 3 nucleotide words of mRNA. And then the mRNA transcribes these little words into amino acids, forming a protein. Okay, so here's an example. I'm going to get a different color here. Let's try green. Let's try that green. Okay, so here's my DNA, and you always have a template. And then the template is transcribed into mRNA. Okay, and you see this A would base pair with the U, the C would base pair with the G, the C would base pair with the G, etc., and so on. And then every 3 bases, okay, is one of our little words, and it corresponds to a codon in the mRNA. Okay, and each codon, each group of 3 letters, 3 bases, corresponds to an amino acid. So UUG is tryptophan, UUU is phenylalanine, GGC is glycine, and UCA is serine. Okay, and this we have a nice table that talks about everything, which codons go with which amino acids, but that's basically how it works. Okay, all right. So during transcription, one of the 2 DNA strands, which is the template strand, provides a template for the sequence of complementary nucleotides in an RNA transcript. Okay, the template strand is always the same strand for a given gene. So here, of course, I have 2 strands, my top strand and my bottom strand, but it's the top strand that is the template. The bottom strand is not the gene, only the top strand, and you have to make sure you read the right strand. Okay, during translation, these base triplets, which again are called codons, are read in a 5' to 3' direction. Okay, so if we go back, here's my 5', here's my 3', so we read this UGG, not the other way around. Okay. This says the same exact thing. Codons are read in a 5' to 3' direction, and each codon specifies the amino acid to be placed in the corresponding position along the polypeptide. So how do we figure out which 3 bases or which codon goes with which amino acids? Okay, there are 64 codons total, right? So if I have 4 bases and I use 3 groups of 3 to represent, 4 raised to the 3rd is 64. Okay, so 64 different codons. Of the 64 codons, 61 code for amino acids. And then there are 3 that are stop codons. And oddly enough, they tell you to stop. There you go. The genetic code is redundant because there are 61 of these code for amino acids, and we only have 20 amino acids. That means that more than one codon may specify a particular amino acid. Okay, and we call this the redundancy in the code. Okay, but no codon specifies more than one amino acid. So let's have a look. Here are a couple of RNA sequences. How do I know it's RNA? Because there are U's instead of T's. So that's how I know it's RNA. Okay. So codons must be read in the correct reading frame. Okay. It means that you must correctly group the codons together by which three bases go together. So let's look at, hey, let's choose purple. So the first one, let's go A, B, and C. The first one, we can group our codons like this, or the codons can be grouped like this, okay? Or the codons can be grouped like this. There we go, okay? And depending on how you group them, you get different sequences of amino acids. So, and the codons, so let's look at this first codon, AUG. So if we go to our handy, oh, look at that. Look, a chart. So our codon, if you remember, was AUG. So our first base is here, the second base is here, and the third base is here. So the first base, you look for an A, there's the A's, and then U from this column, and then G from over here, and you'll see here we get AUG, okay? And this codes for methionine. It's also the start codon, which we'll talk about later. So this would be methionine, okay? CAU is histidine. And then UAG, where's UAG? Oh, UAG is stop. So that's not good. So, but if we group this, so that would be the code for group A. If we look at group B, let's switch to my blue. The group is UGC. So let's see what UGC is. UGC is cysteine. And then AUU, oops, wrong way. AUU is isoleucine. And then AGA is arginine. Okay, so just by shifting the codons by one base, you get two completely different sequences of amino acids. So you have to have them in the correct reading frame. Okay. This chart obviously is, let me erase all of my schmutzy schmutz. Okay, this chart obviously is the key to the genetic code. They come in boxes like this. There's also some that come in a wheel. And I will give you your very own genetic code. So that's exciting. But this is what we use to decipher the code. Okay, little piece of evolutionary interest right here. The genetic code is pretty much universal. Okay, so remember here, AUG encoded for methionine. So in humans, AUG codes for methionine. In monkeys, AUG codes for methionine. In fish, AUG codes for methionine. In corn, AUG codes for methionine. In bacteria, AUG codes for methionine. It's exactly the same. Okay, and so this is kind of cool because it means that if you put a gene from one organism into another, and here we go with our here we go with our transgenic, that G-E-N-I, I seriously cannot spell it all. Transgenic, I did it again. Transgenic organisms. Okay, you put a gene in and it can be transcribed and translated It doesn't matter what species it's in. You can just pop a gene in and it's ready to go. Okay? So that's our kind of a nice evolutionary component for this. And that's it for the genetic code. Okay? On to transcription.